Welcome everyone to this H3D Foundation webinar. We're thrilled to have you. This is, I think, number 6 in our second semester. We're delighted to have, week after week, session after session, some of our favorites joining us again and again. So thank you for doing that. Today, we're very honored to have Professor Rosemary Dorrington from Rhodes University join us. She was requested by our Natural Products team of the GC ADA flagship projects. And so we're so thrilled to have you with us, Rosie. So I'm just going to start by reading her bio to give us a sense and to get everyone up to speed. And then remember, after the presentation, there'll be a chance for Q&A. You're welcome to populate the chat with your questions. Rosemary Dorrington is the Professor of Microbiology in the Department of Biochemistry, Microbiology and Bioinformatics at Rhodes University in Makanda, South Africa. She is also the Principal Investigator of the SA-UK Antimicrobial Drug Discovery Hub. She leads a multidisciplinary marine natural products research group that is active across the broad field of marine biodiscovery, using multiomics approaches to explore the potential of marine natural products as lead compounds for drug development. Rosie, thank you so much for doing this for us and for joining us today. I'm going to hand over to you straight away, and hopefully we'll take some Q&A at the end. Thank you so much.
Thank you for the invitation, and I'm not sure who the audience is and whether you're all chemists or some biologists or a mixture of both. So I'm going to apologize in advance if I oversimplify some of the genomics, but I thought it would be interesting to give the foundation members an opportunity to see something of the biology-based approaches that we're using to find new antimicrobial compounds. So I'm going to just briefly introduce this talk by addressing some ideas of why antibiotics actually exist, and then moving on to how we have developed antimicrobial resistance and the global and economic impact of, and socioeconomic impact of antimicrobial, and why we need new compounds. Then I'm going to talk about the Antimicrobial Drug Discovery Hub, what our objectives are and the kind of work that we've been doing. And finally, I'm going to touch on some of the projects in the lab to give you an idea of the approaches we're taking to look at finding new. So to start with, there's a really interesting article, recent article in Current Opinions that addresses the question, in MBio, that addresses the question of why antibiotics exist. And in nature, the antibiotics have functions that are very different to the functions that we use antibiotics in the clinic for. And those functions, they function usually as signaling molecules, where they regulate gene expression between species and within a species. They can regulate biofilm formation and migration of microbes in the environment. They're often involved in determining virulence, particularly if they are in symbiotic relationships with their hosts. And they're also important in chemical defense, where they regulate the growth and metabolism of microorganisms and larger organisms in their environment. And it's this ability that we have, to some extent, successfully and unsuccessfully harnessed in the clinic. So how they are made or produced in the environment, their biosynthesis. Usually you have a single microbe, a couple of cells that have the capacity to produce the compound that they export into their microenvironment. So they export it into the environment immediately adjacent to them. They produce very small quantities. They only produce these compounds when they need them, because they are energetically expensive to produce. And the producers, if these compounds have antimicrobial activity, the producers have encode what is called self-resistance. They often share that with the microbial species and cells in their environment, the ability to be resistant. And it's this resistance that ends up being the antimicrobial resistance that we pick up in the clinic. So in contrast to their activity in nature, in the clinic, we usually use them to inhibit the growth of a monoculture, so a single isolate or strain of bacteria in very large quantities. We use high doses to saturate the environment to try and kill off as many of the bacteria as possible. And unlike nature, the bacteria that we are targeting, the pathogens, there is often prolonged exposure to the antibiotic, and all of this leads to large-scale resistance in the clinic and in the patient. So the majority of antibiotics that are currently in the clinic are natural products. So a natural product is a molecule that is produced by a living cell. The majority of them are microorganisms, plants or animals. They usually, most of the antibiotics are small molecules, although other natural products can be as large as proteins and enzymes. But in general, the natural product antibiotics are fairly small. And they are usually produced as secondary metabolites. So they are produced as compounds that are not essential to the cell, but enhance the ability of the cell to survive. And you can see from the figure down below that has been generated by the World Health Organization. The majority of antibiotics in the clinic are produced by actinomyces, and a smaller number by bacteria. Fungi also produce antibiotics, most notably penicillin, and then there are an equivalent number of synthetic compounds that have been produced in the chemistry lab. Right, so I'm going to now talk about the global challenge of antimicrobial resistance. It's one of the top 10 global health challenges. In 2019, more than 1 million deaths were directly associated with drug-resistant bacterial infections, and it's expected to double this number without an intervention. This number is expected to double by 2050. And this we don't often think about, but the economic loss due to antimicrobial resistance in terms of additional healthcare costs is estimated to be an annual cost of $1 trillion per year globally. And that is a lot of money. Well, several $1 trillion, but the additional healthcare costs are huge. The main drivers of antimicrobial resistance are the misuse or overuse of antibiotics in humans. And particularly now we are aware of the impact of misuse in agriculture and also animals and plants. And I'll explain briefly now in the next slide or two why this is really important. And then in addition to that, the real problem is access to equitable access to antibiotics and getting the antibiotics in the places where they are needed. The global south is the most affected by antimicrobial resistance, particularly Southeast Asia and sub-Saharan Africa. And the problem is always getting the right antibiotics that are effective to the right place to treat patients. So the mortality rate in the global south is much higher than in the global north. There is a hit list of pathogenic bacteria that's been issued by the World Health Organization. They were originally termed the escape pathogens because of the species that were involved. 2 gram-positive species, Staph aureus and Enterococcus faecalis. And then the big hitters, the gram-negatives, Acinetobacter baumannii, Klebsiella pneumonia, which is the focus of the Gates Foundation call, that was that came out recently, Enterobacter species, including Escherichia coli, and then Pseudomonas aeruginosa. The gram-negatives are on the critical list. The high group includes the gram-positives. And it is worth thinking about the mortality rates due to escape pathogen infections. Antibiotic-sensitive escape pathogens account for a 20% mortality rate, and multi-drug resistant pathogens have an approximately 40% mortality rate. So we tend to talk a lot about the bacterial pathogens, but the real challenge is right on our doorstep, and that is the challenge of pathogenic fungi. In Africa, particularly, is a major problem. Extremely high mortality rate, 70% in parts of Africa, 20% in the US. Azide-resistant Aspergillus species have a mortality rate of between 50 to 90% in immune-compromised patients. And then of particular importance now, and becoming of critical importance, and already appearing in South African hospitals, is multi-drug resistant Candida auris. And to some extent, the thinking is that the rise of drug-resistant Candida auris was fueled by the COVID pandemic, because it is predominantly infecting people who are patients who are intubated on ventilators, and then it is now spread to the neonate ICUs, so particularly preemie babies. And at this point, there aren't any drugs available to treat drug-resistant Candida auris. So the real threat coming down the pipe is fungal pathogens, and there is a real need to start looking for antifungal drugs to deal with these resistant pathogens. Right, so that brings me to the South African, the SA-UK Antimicrobial Drug Discovery Hub. It was launched in 2020 by the UK and South Africa Medical Research Councils to develop an accelerator to develop capacity for antimicrobial drug discovery in South Africa, supported by the UK. It's a multi-institutional program that is now in the an extended phase of funding. It's hosted in South Africa at Rhodes University, and in the UK at the University of Plymouth, where the PI is Professor Matthew Upton. And the aim is to develop a multidisciplinary platform for cutting-edge research in and development of antimicrobial drug discovery platforms to combat and support drug discovery efforts in South Africa and in the region. So the objectives, and this is the overall objective, to develop a pipeline of compounds, head compounds that can be progressed as lead compounds, to assemble a library of natural products, extracts, and pure compound libraries that can be screened and made available to other research groups and research projects. Allied to the physical, chemical libraries, and associated with that, a natural products omics platform that includes genomics and metabolomics data that can be mined in silico. And then finally, to progress, to identify novel compounds that can progress through and into the next phase of research, and to develop the capacity elsewhere in South Africa and in the subcontinent to develop a research network that can take this research work further. So why natural products? I can sense that there are synthetic chemists and inorganic chemists that are feeling a little uneasy because natural products drug discovery is difficult. There are advantages for working with natural products, and that is mainly the chemical diversity of these compounds. And they've been largely underexplored. Many natural products, not all of them, are extremely potent inhibitors of their targets, and they have evolved by natural selection over a long period of time, millions of years, to be extremely selective and highly potent. And then many of them are biosynthetic compounds with enzymes that are selective, and this does help to produce chemistry that is difficult to access synthetically. The disadvantages, the drawbacks, though, is, as I mentioned before, they're often produced in very small amounts, and you have to learn to work with milligram quantities of these compounds as opposed to gram compound quantities. We have, screening for them, screening extracts is difficult and traditionally unreliable, and getting from an extract to an active compound is really difficult. I've dealt with the problem of supply. And then very often these compounds are chemically complex, and they can be difficult to synthesize in the lab. And I'll deal with that again later when I talk about some of the work that we've been doing. So this is the discovery pipeline that we've developed within the AdHub. You'll see that it runs all the way from the ecological, the environment in which the natural products find themselves, through to the field work, and then the collection of repositories of the biological material, voucher samples, DNA, RNA samples, and microbial culture collections, because in many cases, the compounds that we are working with are microbially produced. From there, we generate extract libraries and DNA nucleic extract libraries. The chemical extract libraries are screened for bioactivity on our platform. And from there, we prioritize extracts that have this selected antimicrobial activity for dereplication, the isolation and identification and isolation of active compounds. We have a genomics program that runs in parallel to identify potential biosynthetic pathways. And we're also looking at resistance mechanisms in these biosynthetic pathways. And then from there, the isolated compounds are selected for potential, their hit validation and the potential progression through to leads. So our current bioassay platform, and I think it overlaps a lot with, there's a lot of overlap with H3D. We don't work in general with clinical isolates because we don't have the facilities for that. But we do have an in vivo assay for most of the escape pathogens, including the critical gram-negative isolates. We have some. We have available to us some multi-drug resistance strains, in particular Klebsiella and Acinetobacter baumannii. We also routinely assay for cytotoxicity against Haykin-Heeler cells, and in collaboration with other groups of Rhodes, any cytotoxic extracts are triaged against selected cancer cell lines of importance to the region. And then finally, we have a small recombinant, we have a small library of recombinant strains that we're expanding, that we can use to validate the targets of isolated compounds, most notably by overexpressing the target and then looking to see whether they rescue sensitivity. They rescue sensitivity to the drug. We have a small, we have an in vitro platform, and the focus of the in vitro platform is on 3 enzyme families. The Staph aureus pyruvate kinase, which is involved in glycolysis, the DOX-P reductor isomerase, which is an enzyme involved in the isoprenoid pathway in gram-negatives, and then the HMG CoA reductase, which is the equivalent enzyme in the gram-positive positive metabolism. And then we also have some work that we've been doing on HPPK, which is an early enzyme in folate biosynthesis. I'm not really going to talk about it too much, but we have in vitro assays for selected enzymes from different escape pathogens, so that we can screen broadly against our extracts and then get to the active compounds as quickly as possible. Right, so at the moment, this is a summary of where we are. We have a reasonable collection of marine natural products extracts. On average, we estimate there are between 100 and 200 compounds in each extract, so we're probably looking at between 150 and 200,000 compounds that we can screen there. We have bacterial and fungal culture collections that are currently being extended, and then a collection of synthetic compounds that come out of the compound collections at Rhodes University. We screened a significant number of these extract libraries and cultures, microbial cultures. And we currently are at the point where we've prioritized approximately 50 marine natural product extracts with activity against priority organisms, Klebsiella, Acinetobacter, and E. coli, some Staph aureus, and several of those have activity against our target enzymes. And then we have some fungal extracts and microbial extracts that are also active against target enzymes that we're taking further. Right, so I'm going to talk now about our marine natural products work, mostly because it's what I do, and it's what makes me get up in the morning and makes me smile. And I'm going to talk to you, this is our pilot lab, or our playground, Algoa Bay, which is just off the port of Gqeberha and the Kruger port, also Port Elizabeth. It's an hour and a half's drive down the road, and we have access to ship time, etc. This is known as a marine hotspot, and approximately 20% of all the marine macrofauna that we collect here have yet to be described. So there is a great deal of novelty in terms of species. We look mostly at sponges and ascidians, and the more novel your species are, the better the chances are that you're going to find novelty, chemical novelty. And this is what it looks like when you fly in over Gqeberha. You see the sea, but when you look below, between 20 to 50 meters and down to 100 meters, there is extraordinary diversity. And I never fail to be amazed at what is down there every time we go out into the field. How we do that is through collaboration with the South African Institute of Aquatic Biodiversity and the African Coelacanth Ecosystem Program. They have an infrastructure platform that includes a ship, the Observer, and equipment that enables us to get down into depths that are inaccessible by diving. So start this. This is our ROV, a remote operating vehicle, has a collection arm that can grip and select a sample, and it can then it has cameras, and it's self-propelling, and it's attached by an umbilical cord, and then it brings these samples up to the surface. It's managed by it's operated on board on the boat. Oopsie. And we can then bring up the collection of sponges and ascidians, and they are then transported to the lab where they are processed for the libraries. We take samples for taxonomic identification, sample biopsies, small samples, probably a gram or two, for chemical extraction, and then samples are stashed away for culture, for preservation of bacteria, symbiotic bacteria for later, and DNA and genomics activity. So there is a graphical description. You have your sponges. There is the arm of the ROB. You make the extracts. You then assay them. We use rosazurin assays to assay metabolic activity. From there, we fingerprint the bacteria, and we can do genomics on them, isolate bacterial isolates that we screen for the production of antibiotics. And then of those extracts that are active, then get progressed for mass spectrometry analysis, and the data from the mass spectrometry is analyzed by molecular networking and then prioritized for the isolation and characterization of active compounds. And we can get from the field through to the bioassays and the molecular networking on a good day within 10 days. So within 2 weeks, we can have an idea of whether we are dealing with new compounds, potentially novel scaffolds, and we have a good idea of what the activity is. We will have been able to screen that against our entire in vivo and in vitro bioassay platform. Right, so here's an example. This was a pilot study we did with approximately 200 sponges that we collected between 2022 and 2023. These are the kind of activities against some of the bacteria that we screened. Majority Staph aureus assays. Baumannii seems to be pretty a higher hit rate for Baumannii. Pseudomonas, only a few. And then E. coli and is also pretty recalcitrant, but the hits that we've got have been really interesting to follow. A lot of cytotoxicity, and the cytotoxicity then is compounds that are cytotoxic are usually triaged for other research. And then here I've included the Staph aureus pyruvate kinase because I'm going to talk about it in a little while. So here's a typical molecular network generated by our chemist Jarmo Kalinsky. In this case, he screened 181 sponges. There were almost 12,500 compounds or features that were sorted into clusters of related compounds. And from there, we identified the sponges, the extracts that were active, and prioritized those active extracts for further study. And so on the left here are the extracts that came out of those, out of the 181 extracts. The gray, the clusters, each cluster represents a series of compounds that are structurally related. The black ones are extracts or compounds families that we have been unable to identify. Those that we have been able to identify that are grey, we've actually been able to extract the compounds and confirm the classification of those clusters. And I'm going to talk to those, the specific set, the pyrroluminoquinones here on the right, a family of bis-indole alkaloids called the topsentins. I'll just speak to them briefly there. And then some bifunctional sphingolipids that have come through that we've used as a pilot study to test our discovery pipeline. So the sphingolipids first, they're a set of compounds that were isolated from sponges, a sponge called Oceanopia ramsayi. We first were alerted to these sponges from a library. It was assembled in 1994. It's a 30-year-old library, and the compounds are still active, and we're still finding new compounds of interest in the library. Dele Abdissa extracted these compounds from a couple of specimens that were collected, and we prioritized these compounds because they were very active. In addition to being very active against our panel of bacteria, we were unable to identify any select any resistant mutants, and their time of kill was extremely rapid. However, these compounds are very cytotoxic, and while they may not be suitable for use as antibacterials, we are interested in their targets in the hosts in the bacterial cells, because it may identify new targets that could potentially be screened as targets for the development of new antibacterial compounds. It's interesting, though, what doesn't make it into the antimicrobial activity, further antimicrobial work, does make it into the anti-cancer work, and these compounds are highly selective for certain types of breast cancer cells, most notably recalcitrant triple negative cancer lines, and so they may have application in developing new anti-cancer drugs. This is the second set of compounds that we've worked on. It was actually, these compounds were the first that we identified because they were active against Staph aureus. And when we discovered that they had previously been identified as compounds, the topsentins, in particular bromotoxentin and bromohydroxytopsentin, as compounds that were active against the Staph aureus pyruvate kinase. We re-isolated these and several of the other compounds, related compounds, and assayed them. And in Paul Race's lab in Newcastle, they generated an overexpressing Staph aureus isolate, and showed that these compounds were still toxic and still had antimicrobial activity in the strain overexpressing the enzyme. So while they're active against inhibiting the enzyme in vitro, the target, they probably have off targets in the cell, and the antimicrobial activity is due to activity elsewhere. So these compounds are not useful in terms of pyruvate kinase inhibitors. But the other hits that we've had from the fungi and some of the sponges, in fact, were rescued by overexpressing pyruvate kinase in Staph aureus. So the assumption is that their biological activity targets pyruvate kinase, and those warrant further study to see what they are. We know that they are not topsentins from the molecular networking. Right, so I'm going to talk about my favorite sponges, the trunculate sponge, a family of sponges called the Latrunculidae. They are fondly known as our lab sponges, and that we've done a lot of work on. These are cold water sponges that are particularly abundant in Algoa Bay, and we've been able to work on 8 or 9 species that are endemic and close by. And they produce pyrroluminoquinones, which are alkaloid compounds that are traditionally isolated as anti-cancer compounds that target topoisomerases. We've been interested in them from a laboratory point of view to develop new approaches to drug discovery work, but also because it appears to be a versatile scaffold that has potentially many different targets. So this is Jarmo Kolinsky's PhD project, where he characterized the compounds, used molecular mass spectrometry and molecular networking to characterize compounds from multiple species of these sponges, and identified more than 200 in those extracts that are all chemically related to each other, but that potentially can be accessed for screening for drug development, or by screening the different compounds. In general, there are 3 classes: the more simple maciluvamines, tizicamamines, which are believed to be derived from the macrolidamines, and then the discorabdins. So, and here the compounds, we've isolated several of these. Here are 11 of the compounds that are more purified and isolated. We've assayed them against, some of them have antibacterial activity at MICs below 20 micromolar. Most of them are extremely cytotoxic, but not all. And many of them are active against mammalian topoisomerases, but thus far we haven't been able to show that they're active against bacterial topoisomerase or gyrase. And the reason we are interested in these at the moment is that they have come up as being selectively active against Acinetobacter baumannii, and in particular, they inhibit in vitro, the same extracts inhibit the Baumannii DXR enzyme. So there's a potential that they may be targeting isoprenoid synthesis in this organism. So we are following that further, and we are looking at doing in silico structure function and drug structure binding modeling to see whether to find the how these compounds interact with the Baumannii, and then to map them against the other enzymes that we have, DXR enzymes, to begin to understand what the specificity is. So not all of these compounds are cytotoxic, and I don't think they're going to end up in the clinic, but what they can do is give us an idea of what, through their interaction with the DXR enzymes, of new areas on the enzyme that we can put binding domains that inhibit the enzyme that we can potentially target for in silico screening of scaffolds to develop new compounds. I'm going to finish off by just talking briefly about the genomics work we've been doing. And we know we now have data, in general, many of the compounds that are produced are microbially produced. You can't go out into the field and harvest 20 tons of these sponges or any of your marine macrofauna that produce these compounds to isolate enough compound to be able to get into clinical trials. So it's really important that you get from the structure to either the synthesis of the compound in the synthetic synthesis in the chemistry lab, or alternatively explore biological synthesis by employing either the host bacterium that originally makes the compound or generating recombinant methodologies for synthesizing by fermentation, or a combination of both, using enzymes that are key in the biosynthetic process and expressed in E. coli, and combining them with synthesis to develop an efficient synthetic process. So here's an example of this. This is Gabby Solomon's PhD project, where she's hunting the pyrroluminoquinone production pathway. We've identified it in the sponges, or we think we have. This is a pathway, a hybrid non-ribosomal peptide synthase, polyketide synthase pathway that we've identified in several of our sponges, and it's predicted to produce macrolidamine A. And when we screened the libraries of culture libraries and their genomes, we discovered two related bacteria that are culturable that have the same genome, the same biosynthetic gene cluster. And Gabby is now looking to confirm whether this gene cluster A produces macrolidamine. And if it does, we're looking to see how we can, whether we can produce sufficient quantities in the bacteria, or alternatively, whether we can use some of these enzymes to tailor these scaffolds to enhance selectivity and potentially develop new targets for drug development. And finally, I'm going to give you something very complicated, because the same approach we're using for finding microbial compounds. So these are a set of 8 bacterial isolates that were isolated from stromatolites in the Eastern Cape. They have antimicrobial activity, and they are particularly rich in the potential to produce compounds. So on the left here are the biosynthetic gene clusters. This is Idris and Janji's work. And the next slide shows you the metabolomics. So this is what they can potentially produce. And on the right are the compounds that we've detected by in mass spectrometry analysis of extracts from these bacteria. And we have been able to start identifying or matching the compounds that we've identified in the extracts with the biosynthetic pathways on the bacteria. And also we're hoping that we're going to be able to identify or begin to work on some of the biosynthetic pathways that have potentially novel compounds. Most notably, what was discovered in 2 of the strands is an antibiotic called actinomycin D. Actinomycin D inhibits Staph aureus activity, but it is an extremely potent anti-cancer drug. So it was good to be able to validate the integration of these compounds, of the work that we're doing, the genomics and the metabolomics data. You're only as good as the people you work with. On the left-hand side is a slightly dated a group photograph with our collaborators Sam Waterworth and Shirley Parker-Nance. Shirley Parker-Nance, one of the most important people in the marine natural products work. She's the marine biologist. She's our taxonomic guru, and she has a remarkable instinct for finding sponges and ascidians with really interesting biological activity. On the right-hand side is a photograph taken at a recent meeting of the AdHub collaborating team. Karen Jacobs. Karen Jacobs is from Stellenbosch University. She's leading an antifungal drug discovery program, a new antifungal drug discovery program at Stellenbosch. We have He Deng. He is at the University of Aberdeen. He is working with us on developing the microbial natural products discovery. He's an expert in actinobacterial pathways. Paul Race, I mentioned him earlier. Next is Paul Race from Newcastle, who's been working on the Staph aureus pyruvate kinase and looking at structure function, matching the compounds with the protein structures. My mouse has almost disappeared. This is Matt Upton from the University of Plymouth, my co-PI on the project, and Rachel Goss, who is at the University of St Andrews, who has been really important in helping us pull all the networks together. And I'm going to finish with this. If you are not already aware, we're hosting the Bio26 conference in Mukunda from the 26th to the 29th of January. There is a very strong drug discovery theme to the conference. The abstracts, unfortunately, the call for abstracts is now closed, but registration is still open. And we will be running a metabolomics workshop at the conference on Friday, the 30th of January, followed by hands-on mass spectrometry training in our facility the following week. So if anybody is interested in joining the conference, please log on to the conference website or contact me about attending the workshop and or the training workshop the first week in February. Thank you for your time.
Rosemary, thank you so much for that really thorough and insightful presentation on the work that you and your team have been doing. It seems like quite an extraordinary team you've got grouped together there, and a real powerhouse of expertise. And I'm just wondering, and guys, please raise your hands if you want to ask questions, but I'll kick it off. if everything goes really perfectly, like best case scenario for the ADD hub, what would happen next? What would we next hear from you? What would be the best case scenario?
We are following some hits that we think are novel, and we are in the process of doing the structural work, identifying them and doing the structural work. And so our real goal is to have some compounds that can be progressed, validated and progressed as leads at the end of, well, not at the end, but that's our next big goal. We're still in the early discovery phase. And in 2020, we thought we'd get to leads within 3 years, and that's really not going to happen in this field. We're also now beginning to look at taking some of these projects and getting them funded to do the lead, to progress them through to leads. But I think what's really important is to develop a research network. We're looking at one of the big concerns, and I think all the project leaders that are potentially that will know this, is sustainability. You kind of lurch from one 3-year project to another, and it's really important that whatever capacity is being developed during these projects is retained and developed further. So your question, what should you hear from us? I think the most important thing is going to be that we can sustain and develop the work that we've done further, and develop it as a hub that can support research elsewhere in the region and in Africa. Big, hairy, audacious goals.
They sound great. I'm going to send all the manifesting energy I can to you for those. Sizwe, over to you. You can unmute yourself. Go for it.
Good afternoon, Prof. Thanks for the nice presentation. I just want to ask if, because I'm also interested in this kind of work. So we're looking at compounds from fungi, and we have been testing them against HIV and TB. So we had the same thought process as what you have just explained, of looking at linking the metabolites with the biosynthetic genes, and we have started on the omics, on the combined omics, but it is really struggling because you have to have so many expertise, like a combined expertise, like omics, like your metabolomics, your transcriptomics, and your genomics. So we still trying to get our expertise in one lab, but it's very difficult. So I wanted to ask if it's possible for us to take along with your group, so that we can maybe learn in the process. I don't know if that is possible.
In fact, it's not just possible, it's really important, and I should emphasize that more. Our role as a hub is to support researchers outside of the hub, and to develop that network further. So we would certainly, it would be really, really nice to be able to collaborate with you, and then also to put you in touch with the Stellenbosch group. Which university are you at, Sizwe?
I'm at Northwest University.
I think it would be really good to put you in touch with Professor Jacobs's group as well. And we certainly could be running some genomics workshops. We have a pipeline, it's mostly bacterial, but we do have a pipeline for assembling genomic sequencing. The other thing that I did forget to mention is our bioassay platform and our metabolomics platform are available for use to provide a service for other research groups. So if you have extracts that you'd like to have screened against our bioassay platform, we would be very happy to screen them for you. And also, we're now in the process. In January, we will be reopening our mass spectrometry unit and making it available for screening metabolites and for the type of work that we're doing. Our aim is to keep the cost of using these facilities as low as possible so that they are accessible to research groups in the country.
Okay, thank you. I will follow for further engagement. I will email you so that you can take it from there.
Yeah, absolutely. It sounds very exciting. Really interested to hear about what you are doing.
Oh, thank you so much.
Amazing. I can see a collaboration seed was just sown here. Don't let it be forgotten, the H3D Foundation webinar. If there are no further questions, I will just say a big thank you to all our participants for joining us today and for giving us your attention and making it worthwhile for such esteemed guests to come on and share their knowledge. And then a huge thank you to Rosie. Thank you so much for giving us your time and expertise and knowledge, and also for being so open. I mean, it's so clear that this work has to be done in teams. We've got to collaborate, we've got to work together, and yet it's sometimes really difficult to do that for so many reasons baked into the scientific process. And so I can really tell that in the ADD hub, you are really working at how do we actually get to the end goal, not how do we retain all our expertise just for us. And that's really, really welcome news, I think, especially on the African continent. So thank you for that.
Thanks. Yeah, I think that's really important. We have to be able to develop our own capacity, and there aren't enough of us to be able to do it by ourselves. And we're much stronger if there are multiple groups all working together.
Thank you so much. Guys, I've populated the chat with our feedback form where you can suggest topics and give us some feedback as well. Of course, all our H3D Foundation links. And if you want to get in touch with Prof. Dorrington, you can always email me or find her online as well. So with that, thank you so much, everybody, and we'll see you next time.
Bye.
Bye.